Good day, grade 11 learners. Welcome to today's life science lesson. My name is Susan Wiese. This lesson is brought to you by Gauteng Department of Education in collaboration with Sibona Discovery Centre and broadcast by Pakama Research and Development. Today we're going to look at the introduction to evolution. And I know you're immediately going to say, no, lady, we didn't do this in grade 11 this year. I know you had no chapter called evolution, but in grade 10, you already gained some valuable information and knowledge that's going to help you in grade 12 with evolution, as well in grade 11. So today's lesson, we're going to look at what is the things that at this point in time you need to know and understand of evolution. So let's start the lesson. First of all, what is evolution? Evolution is the process of change in the genes that have transformed life on Earth from its earliest forms to the vast diversity that characterizes life on Earth today. So evolution is all about changes that is happening. Biological evolution means all present-day forms of life have descended from and are related to those that lived in the past and may look different because they became modified from one generation to another. If you look at my diagrams of the horse, you will see that the horse that we know today is looking different from the horse that existed millions of years ago. And this is the very first horse that we know of that lived on Earth. And can you see there was a change over millions of years? And that is why we are saying evolution is the change that is happening in organisms over millions of years. Yes, sometimes it does happen very fast, so that what we have today is looking different from what was existing millions of years ago. We're first going to look at how do we know that evolution is taking place? What evidence do we have that evolution is taking place? And you will see that there are mainly four things that we are looking at. First of all, we're going to look at biogeography. Now, what is biogeography? That is really how animals that are the same are dispersed over the world. All right, and you did this when you did grade 10 life sciences. All right, so we're just going to refresh your mind a little bit. The second evidence that we have and that we look at is that of genetics. And I know at this stage you do not know much about genetics. And that is why next year in grade 12, you are definitely going to stand still at DNA and RNA and genetics and heredity. The third one that we are looking at, and that's evidence for evolution, is homologous structures. You have also looked at this a little bit in the past 2 years. And then the last one, the biggest one, is the fossil record. And in grade 10, you looked at fossil formation, at fossil dating. So we are just going to refresh your mind and make sure that all the background information that you are needing for next year's grade 12 evolution section, you have, and that you have it fresh in your mind. Let's start with the first evidence, biogeography. Now, 250 million years ago, that's a long, long time ago, the Earth was basically one big continent that was called Pangaea. Now, that was in the Permian period. If you remember your time scales that you did in grade 10, you will remember this period. All the continents were joined to form one landmass. So it was a very big continent. Animals could move across this large landmass. So animals from the north could, if they want to, move all the way to the south, if they want to. Plants could also spread across the landmass because there was nothing that was stopping them, that was blocking them to go from north to south or south to north. All right, so this is how Earth looked like about 250 million years ago. Then if we look at about 200 million years ago, the continent basically split in two, and they start moving away from one another, one piece to the north, one piece to the south. And that was in the Triassic period. Now the piece that moved to the north was called Laurasia. And the piece that moved to the south was known as Gondwanaland. So, and so we had, at this period, we had basically two land masses, one to the north, one to the south. Animals and plants on the northern land mass were separated from those in the south. So now suddenly what happened is, if you were in the north, if an animal or a plant was here in Laurasia, they could actually not move to Gondwanaland. And the same for the plants and the animals in the south, in Gondwanaland, they couldn't really move to the north because now they were separated from one another. And then, about 65 million years ago, we in the Cretaceous period, we see that those two, the north and the south land masses, now start to spread also to the west and to the east, and then we see the land or the continents in the position more or less like we know them today. And what is important is, during the movement of continents, some parts sank below the oceans and other parts rose up to form mountains. So if you now try and you want to reverse this process, and you want to say, let me take the parts from the east and the west and pull them together, and I take the parts from the north and the south and I put them together, I must basically find Pangaea again. You'll see it doesn't fit 100% because some parts actually sank below the ocean, and then other parts then suddenly have mountains that wasn't there before. All right, so this you did in grade 10. So it's very important for you to remember this, because if you understand this, you will understand the biogeography of our animals and plants. Now, if we look in total, you will see that first of all, we started off with one big landmass that was known as Pangaea. And then there was a split towards the north and the south, and we have Laurasia, that's basically that part, and we have Gondwanaland, that is the bottom part. And then later, about 65 million years ago, the continents actually split further. They moved away to the east and the west, and this is now how we know basically the modern world of today. Now, what influence or what evidence does this give to evolution? The evidence we have is, if we now look at animals that are more or less the same, like for instance, the flightless birds that we found in the southern hemisphere, you will see that in Africa, we all know the ostrich. All right, so in Africa, we know the ostrich. In South America, they have a rhea. Can you see that it looks very much like a niece or a cousin of the ostrich? And then here in New Zealand, a little bit further away, we have a kiwi, also flightless bird, a little bit smaller, not as tall as the ostrich. In Australia, we've got an emu, and then also in New Guinea, we have the cassowary. All right. So if you look at these birds, they all look the same, more or less. So it looks like somewhere, somehow, there could have been an ancestor. And because of what happened over these millions of years, when the continent actually split up into pieces, these birds landed up in different parts of the world. And because they moved, that means that the environment of these continents changed, because some of them moved closer to the North Pole, some of them closer to the South Pole, and some of them even closer to the equator, where we all know it's much, much hotter. So what we have is the effect of continental drift on biogeography is it provides us evidence for the evolution of ancient species. So it shows us that somewhere millions of years ago, that these animals might have been, only there was one of them. We call that the common ancestor. And because the continents split and moved away from one another, these animals actually got separated from one another. And because the environments were so different, they adapted to the environment and they changed. And therefore today, if we look at them, we can see, you know what, they look like family, but there are changes in them. And this is how we then can explain the fact that there was actually evolution happening in this family of birds. Now, another example is, if we look at the world map that we have now, the present-day distribution of 3 closely related species of the dog family. The coyote, can you see? And this is a coyote here. The jackal, there we all know the jackal that we found even in South Africa. And the dingo, there is the dingo. If you look at the photos of these 3 dogs that are from the dog family, they look very much the same, yet there are differences. So how do we explain this? How is it that these 3 dogs, or dog families, species of dogs, look so much the same, but yet they are in so far apart on Earth? There's no way that they can actually get to one another. And the only way we can explain this is to say that, you know what, many years ago, remember when we had Pangea, when there was one landmass, there was one ancestor. And because of the continents that moved away, or continental drift, the original population actually got separated. And because of the fact that they now they have different environments, they adapted to their environment, and that is why today they look familiar, but there are differences. So that is why we are saying that biogeography can be used to give us evidence that evolution is taking place. Now, if we look at this world map showing us the Earth, you'll see that there are changes. Now, if we look at this question, they say study the diagrams below and answer the questions that follow. And please remember, and I say this every time, before you even look at the questions, do what the question is saying. And the question is saying, study the diagrams. So before we look at the questions, we must first study the diagrams. So if we look at these diagrams, there's an earth ball at position A, then there's one at B and at C. And if you look at them, you can see clearly. In A, what do we have? One big landmass. So there's one big landmass there. And remember, what did we call that? Pangea. And then if you look at number B, can you see that there's a split towards the north and towards the south? And remember, what did we say? The north was called what? Laurasia. And the south, basically this part, was called Gondwana land. And now if we look at C, this is more or less the world map that we know today. Now you go to the questions. You first look at the diagram. You see, what can I observe? What do I see? And then you go to your questions. Now, question 1 is saying, identify the geological process that took place from A to C. So they're asking you, what's happening here? What do we call this? And this is what? Continental drift. Sorry, let me just go back. All right, so this is continental drift. Then, this thing is now jumping around this morning. All right, then number 2, briefly describe the process identified in question 1. So now, if we have said that this is continental drift, they want to know from us, so what's happening here? Why is this happening? And remember, the tectonic plates of the earth are actually moving all the time. And why is that? Because they are resting on the core of the earth. And because that is moving, we get that these plates are moving. And because they're moving, and when they move away from one another, that gap is filled by the ocean. And that means then that the pieces that have moved away from one another is actually now separated from one another. Question number 3, name A, the supercontinent represented in diagram A. We've already said that. Remember when we looked at our diagram, we said, oh yeah, this is when the earth was actually one piece, one land. And that is called, can you remember? Pangaea. All right, so then 3B, the two continents, so now you must give two answers that resulted when the supercontinent mentioned in question 3A broke into two. So they want to know from you, what is this piece called and what is the bottom piece called? Remember, the piece in the north is called Laurasia, and the piece in the south was called Gondwana land. Now, if you're not sure, don't write more than two names, because the question is saying you must give two, and it's even in capital letters. When we mark your questions, and now you've got 3 words there, because you're not 100% sure, is one of them not maybe Pangaea? So now you write three names. You know what we do as markers? We only mark your number 1 and number 2 answer. Number 3 we don't even look at. Even if your number 2 and 3 are the correct ones, you're not going to get a mark for number 3. Because what did the question say? The question said, give the two names. Right. So remember this when you are actually in the exam situation, that you follow the instructions of the question. All right. The second evidence that we have for evolution is genetics. And I know of genetics you don't know much. Don't worry, next year you are definitely going to look at DNA and you're going to look at RNA. But it's mainly DNA that we use, because mitochondrial DNA we can trace back to Eve. And the reason for that is it is your mitochondrial DNA you only inherit from your mother. You can't inherit any mitochondrial DNA from your father, because when the sperm cell and the ovum actually fuses, you will learn this next year. Only the head of the sperm is actually fusing, and the nucleus, in fact, is fusing with the ovum. So the mitochondria that is in the middle piece of the sperm cell is not going to the ovum, and therefore they use the mitochondrial DNA to trace back our ancestors and the ones, well, I want to say the female ancestors. But don't worry about this. This is going to be part of your grade 12 life sciences here. All right. The third evidence we have is homologous structures. Now, if you see the word homologous, homo means what? It means the same. So it's structures that are the same. How are they the same? As similar organisms adapted to different environments, the function of their appendages changed. Result of descent with modification from a common ancestor. So we use this. We look at the skeleton and we see, all right, if I look, the skeleton of a bat, or there's a bat, or I look at the skeleton of a cat, I see some similarities, but yet they don't do the same thing with that part of their bodies. A bird's arm became wings to flap, all right? A human's arm is used to pick up and carry and grasp, etc., things like that. We use our arms to do that. A horse's arm became a foreleg to run on. But if you look at the structure of those limbs, they look very much the same. The fact that all mammals have the same pentadactyl limb structure suggests that all mammals descended or originated from a common ancestor. So that is what we are actually getting from homologous structures, and that the limbs have been modified over time in the different species. Modification of skeletons by descent is therefore evidence to support the theory of evolution. All right, so that's why we say we can use this as evidence to show that evolution has taken place. If we look at this question, all right, the diagram below shows the structure of the forelimbs in different species. Now we have man, so that's human. There's a cow, a horse, a whale, and a bird. All right, can you see all of them have actually 5 structures or 5 pieces that looks the same? Now they're asking us in the questions, name the line of evidence for evolution represented by the diagram. All right, because they show you that there are, and they've actually highlighted some of it. It means that you must immediately know it's the same, so it's homologous structures. All right, you can also call that modification by descent. And then in question 2, they're asking us, describe how the line of evidence mentioned in question 1 supports the theory of evolution. All right, so now you must describe it. And how will you describe this? What will we say? Although different species have a similar bone structure or a body plan, these structures are adapted to perform different functions. So they have a similar structure, indicating then that somewhere, somehow, millions of years ago, there was a common ancestor. All right. So this is the third line of evidence that we have to prove that evolution is taking place. So the 4th one that we have is the fossil record. And in grade 10, you have studied fossils. And remember in grade 10, you actually said that if we look at rocks, we know that the rock deepest in the earth is older than the rocks towards the surface of the earth. So that means if we find fossils deeper down, that fossil is much older than a fossil that we find near the surface of the earth. So remember, the oldest rock strata are found deepest in the earth, while the youngest ones are occurring near the surface of the earth. That you studied in grade 10. You must remember this. Fossils of the organisms found in the deepest strata therefore lived on earth much longer ago than those species which are fossilized near the surface. Right, so the deeper down you dig up this fossil, you will know that that fossil is actually older than the one that you find close to the surface of the earth. And what are fossils? I know if we think of fossils, we only think of skeletons. We think fossils are skeletons. No, there are different types of fossils, in fact. You get a mold or a cast fossil. Like for instance, if you look at this photo here, you will see that these are actually footprints. So here are footprints that was made in stone. It could be that there was a volcano eruption and somebody walked over this, and that is why we see these footprints. So that's a type of a fossil. Then we also get preservation in natural matter. And the photo here is showing a fly. Can you see there's a fly there that is caught in amber. All right, so that's also a fossil. And then the fossil that we always think of, the skeletons, is mineralized fossils. All right, and then today we also have living fossils. We have fossils that existed and lived on Earth millions of years ago, and they're still with us. And one of them is a coelacanth. And this one you do find at the coast of South Africa as well. All right, so this is just the kind of fossils. So when we say fossils is evidence for evolution, don't just think of a skeleton, because we're talking here of much more than just a skeleton. Now, what is important for you? You have studied the skeleton of humans. And how is that important then in human evolution? We're going to look at certain structures and certain features that you must be familiar with. I know you've studied all the names of the bones in your arm and in your fingers and the bones in your legs. And you looked at the skull and you look at the vertebrae. So that is background information, but you're not going to need those names so much next year. But there are certain ones that is very, very important. So now look at them. Let us look at them. The first one is when you're going to study the differences between African apes and humans, you're going to look at bipedalism. Now, what is bipedalism? Bi means two. So it means it's animals that walks on two limbs, like us. We are bipedal. Now, what is important here is to look at the position of the foramen magnum. Do you know where your foramen magnum is? You have one. You definitely have one. Where's the foramen magnum? At the bottom of your skull. All right. If we look at the photos here, it is that hole that you will find at the bottom of the skull. Why is there a hole? Remember, your brain is where? Your brain is in your skull. And from your brain, you have a spinal cord that moves down the vertebrae column. And that spinal cord, one way or another, it must be able to get from the brain into that vertebral column. And that is the opening, the foramen magnum. All right, now the position of that foramen magnum in the skull is very, very important. It tells us something. In humans, it's in a more forward position. So it's moving, if you turn the skull around, it is in a more forward position, as here in position number 4. And then in less evolved animals, we will see that actually that position of the foramen magnum is moving backwards. So it's in a more backward position of the skull. You cannot say it's underneath the skull. They're all underneath the skull. You cannot say it's in the middle of the skull. All right. You must say, when you look at the position of the foramen magnum, you must say in humans like us, it's in a more forward position because we're bipedal. And then in African apes, it's in a more backward position because they are quadrupeds. So they walk on 4 legs. All right. The next thing, all right, here just to show is, can you see that with us, it goes, that's where your spinal cord is entering your skull. And if you look at apes, can you see, because they're bipedal, it's entering there. So there is a difference. Then the next thing we're looking at is the spine. The form of the spine also tells us something. And what you must know, your own spine, if you take a photograph of your spine, you will see that your spine is more S-shaped. It looks like this. While in African apes, the spine is more C-shaped or curved. All right. Why is that? Because our spines are S-shaped, it helps actually to absorb shock when we walk. And it also allows us to have more easily a way of walking than what we find in African apes. So you must know that your spine, what is the form of your spine in humans, you must know it's S-shape, while in African apes, it's C-shape. Then the next one is the pelvic girdle. All right, I hope you know where your pelvic girdle is, and I think that the 2 line diagrams actually show you clearly where your pelvic girdle is. If we look at the pelvic girdle of a human, and we look at the pelvic girdle of an African ape, humans, it's short and wide. If you look at the red lines here, there's the short, all right, and there's the wide. Compare that with that the pelvic girdle of an African ape. And if you look at that, you'll see an African ape has a much longer but a much narrower pelvic girdle. And the reason for that is, once again, because we are bipedal, it helps us because we're bipedal and they are quadrupeds, so they walk on 4 legs. Then brain size. Now I know, and now you must listen very carefully. There's a difference between a skull and a cranium. What is a skull? It's all the bones that is in your head. That's your skull. But where's the cranium of the brain? The cranium is only this part where the brain are actually situated. That's your cranium. So please, you must know the difference. So if somebody is talking about the skull, they're looking at all the bones. All right, your cheekbones, everything that's in your skull. But if they're referring to the cranium, they're only talking basically at this part, because that's where your brain are situated. Now, if we look at animals that are more evolved, their cranium is bigger than animals that are less evolved, because the size of your brain is determining your intelligence. All right. So in more involved animals, you get a bigger cranium. In smaller or less involved animals, you get a smaller cranium because they have a smaller brain. Now, no, don't be misled. If you look at the size of the brain of a whale, it's much bigger than the size of the brain of a human. But yet, we are more intelligent than whales. It's also in relationship with the body size. All right, so although we're looking at the cranium size, you must also put that into relationship with the body size. So yes, the cranium or the brain size of a whale is much bigger than that of a human, but be more intelligent. Because if you work out a ratio, you will see that the ratio between the brain size of a whale and the body size of a whale is much smaller than that of a human. Then palate shape. I hope you know where your palate is. And if we compare that, easy. Humans, we can see it's a nice C shape. There's our palates. And if we look at African apes, and there's a photo of a chimpanzee and a gorilla's palate, it is much more U-shape or rectangular. All right. And humans have got a smaller palate and a jaw, while African apes have got a bigger palate and a jaw. All right. Now, the next one is dentition. You have studied this. You have studied the different types of teeth and how they look in the human. And what is important here is to look at the different types. You will see that if we look at a gorilla, what is very big in a gorilla, which you can see on this photo very clearly, is the canines. All right. Our canines are actually very small. The reason for that is gorillas are still tearing their food. We don't do that because we have evolved. We now cook our food. We don't have to tear the food like the way that they do. All right, and they also use that to kill. So important. And then furthermore, look at the opening or the gaps in between the teeth of the gorilla. Big. In humans, we have no gaps or very small gaps, and that's also a evidence that we use when we look at skeletons to see how is this a more evolved animal or is it a less evolved animal. Prognathous means having a projecting lower jaw. All right, and I think that's easy. If you look at humans, we actually have flat faces, but if we look at the gorilla, because of the jaw of the gorilla, the lower jaw that is so big and extended, we can see that in them you get actually a slope face, like you can see in this diagram. African apes have bigger jaws. Humans have a flatter face than African apes, and then humans have a chin. African apes do not have a chin, or it's poorly developed. Can you see if you look at the photo here? This poor guy, there's no chin. But in humans, there's a very nice developed chin. All right, that your mother can actually grab and shake your head if you don't want to listen to her. Cranial ridges. If you're more evolved, you have lost actually your cranial ridge. I want you to feel on top of your skull. Do you feel bones sticking out there? No, there's nothing there. But if you look at the skull of a gorilla or a chimpanzee, you can clearly see that they have this ridge there. Can you see there on the top of their skulls? And the reason for that is where the muscles that are actually attached to the jaws that is where they are fixed. And the reason for that is, remember, to be able to tear that meat, you must have very strong muscles that's connected to your jaws. And we don't do that because we eating cooked food, so we don't need those very strong muscles anymore. So as years have passed, we don't have this anymore, this cranial ridge. And then also zygomatic arcs or arches, that's basically here in the skull. And humans, we don't have that anymore because once again, that is also where muscles are attached that helps to move the jaws. So we don't have that. Brow ridges, it's easy. Feel here, just above your eyes, you will feel it's actually nice and rounded. All right, and it's not sticking out like here. But if you look at the brow ridges that we find, for instance, in a gorilla, you can clearly see here, they're very, very big. All right, so the lesser the brow ridges are basically extended, the more evolved you are. And now let's quickly look at a question. In a study to establish the mode of locomotion of some species, scientists compared the pelvic structure of their fossils. They established that one of the two species were bipedal. What does bipedal mean? By two. So that you're walking on only two limbs. The diagrams A and B below show the pelvic structure of the two species drawn to scale. All right, so they're drawn to scale. Now let's look at the questions. Which diagram, A or B, represents the pelvis of the organism that was bipedal? All right, remember, bipedal is humans. This is the pelvis of a human. Because why? It's short, but it is wide. All right. This one is the one of the African ape because it's long and it's narrow. If you look here, narrow. Explain your answer to question 1. I've just actually did that. Explain how the spine of the organism represented by B, so B, we have said this is a human, have contributed to its mode of locomotion. All right, explain. So you must have a statement and then you must clarify that statement. First of all, what is the shape of a human spine? Remember, we looked at that. It's what shape? It is S shape. And the reason for that is that shock absorber, and it makes the human bipedal. Then describe one difference in the position of the foramen magnum between organisms A and B. If we say B is a human, where's our foramen magnum? It's in a more forward position at the bottom of the skull. And in A, it is in a more backward position at the bottom of the skull. And with that, we have come to the end of the first 2 sections that we have to discuss today. We're going to take a short break now of 5 minutes. Don't run away. Just get up, stretch your legs a bit, and in 5 minutes' time, we're going to start with the interpretation of a phylogenetic tree. Now, if you were asleep this morning, you better wake up now, because this is a skill that you have to master before you get into grade 12. So if you don't know what I'm talking about or how to interpret this tree, it's now the time to make sure the adrenaline is flowing in your body, that your ears are open, and that you listen very carefully. So in 5 minutes' time, we're going to resume our lesson.
Good day. My name is Vedika Maraj and I am one of the subject advisors for life sciences in Gauteng. Today I'm going to be teaching you how to interpret phylogenetic trees and discussing evolution in present times. So, without further ado, let's start. Let's recap from grade 10. What do you remember about scientific names? If you will remember, if we go back to grade 10, you'll remember that our scientific name is broken up into two parts. The first word is the genus, and the second word is the species word. And if you look at your textbooks and you look at your question papers, you will see that when the scientific name is written, it's written in italics, like this. Let's just recap there again. Homo in this instance would be the genus name, and sapiens in this instance would be the species. Please, I also want to draw your attention to the first word, which is the first letter of the first word is capitalized, and the first letter of the second word is a small letter. Now, you can't write in italics. So when you are writing in exams or you're writing in your workbooks and you want to write a scientific name, how would you write it? You would write it like this. As you can see, the words are underlined. Both words are underlined. The first letter of the first word is capitalized, and the first letter of the second word is in small letters. So this is how we want you to practice writing. So when you come to grade 12 next year and you're writing your examinations, this is how you will write the scientific names. I want to draw your attention to this word over here. So genus, you know, right, from grade 10. And genus is the singular of plural. Plural is the, of genera. Genera is the plural of genus. So I want you to keep that word genera in the back of your mind because later on I'm going to be discussing it when we're going to do an activity. So what are phylogenetic trees? I've got some nice definitions for you here, 2 definitions that you need to know. A phylogenetic tree is a diagrammatic representation of the evolutionary relationships between species. Another definition is that it's a schematic form that shows the evolutionary relationships within a set of organisms or a group of organisms. So it's basically just showing us the evolutionary relationships between organisms or species. Let's unpack this word, phylogenetic. It sounds a bit heavy, but let's break it up. Phylo refers to the organism's phylum group, and genetic refers to it's from the genes or relationship between the genes. Now, phylogenetic must not be confused with pedigree diagrams, which is genetic relationships between close family members. For example, a family tree. Now, I know you don't know this word yet, but in grade 12, when we do genetics, you will be learning about pedigree diagrams. And just keep the word in the back of your mind. So when you're approaching this topic next year, you will not confuse it with phylogenetic trees. I'm going to go into phylogenetic trees now, and I'm going to explain it to you using a simple diagram at first, and then we're going to go into it in more detail. But as Susan mentioned earlier, I want to re-emphasize the importance of this topic. This is a skill that you must be able to do, and you must be able to master. Because it's not a topic that we can tell you, this is what we are going to ask you. They can ask you anything in a phylogenetic tree. It can be plants, it can be different species, it can be flightless birds. But as if you have the skill of how to approach this and how to interpret and analyze it, then you'll be able to answer any question. So it's marks that you should not throw away. Right, so let's have a look at this. When you have a phylogenetic tree in this way, so you could say it's in a horizontal view. Let's see what it says here at the bottom. On this side, we are reading from left to right, okay? So we have the ancestor, and we are moving to the present day species. So these over here are the present day species, and on this side we have the ancestor. So let's start at this first point. This first point over here, this is the most recent common ancestor of A, B, C, D, and E. Then what happens is the organism evolved, and we have a branching occurring here, branching into different organisms. They are evolving, they're becoming different, and they're changing, which is basic in essential fact evolution. So they are branching off until we reach a new common ancestor at this point. And from there, they evolve again into their descendants. Then we reach the most common ancestor again, but this common ancestor at this point, before the next branch, is the most recent common ancestor of A and B. And at this point, we have the most recent common ancestor of C and D. E had already branched off from the first common ancestor. So I want to explain this again because this is the crux of the whole thing. Okay, it gets a bit complicated, but this is the basics that you need to understand. We're reading from left to right. The left being the ancestor going through to the present times, the present day species. In this instance, it is A, B, C, D, and E. Starting here with the most recent common ancestor for A, B, C, D, and E. They branch because they're going to evolve, they're going to change, they're going to develop into new species, and we're going to get a new common ancestor, which is going to give rise to their descendants. And then this common ancestor over here is the most recent common ancestor for A and B, and this one over here, the most recent common ancestor for C and D. Let's look at a different one. This one is a little bit different because we are reading it from the bottom up. Our most recent common ancestor over here is at point 1. The first branch is at number 1. That is the common ancestor for all the species in this phylogenetic tree. Okay, so it's the common ancestor for A, B, C, D, E, F, G, and H. Let us look at the first branch. So we're going to be looking at this branch over here. A and B and C have a common ancestor at point 2. A, B, and C have a common ancestor at point 2. B and C have a common ancestor at point 3. And this also leads us to say that B and C are more commonly related compared to, or more closely related rather, to each other compared to A, because they share the most recent common ancestor. A is sharing a common ancestor further down, and they didn't evolve again. But at point 3, we have the most recent common ancestor of B and C, so they are more closely related compared to A. And if we look at something else that we can analyze from this, A, B, and C are more closely related to each other than to D, E, F, G, and H. So this branch is more closely related compared to the second branch. Okay, I think I need to go through this one again. So I'm going to quickly erase this, and we're going to do it again. It's very important, as I explained, we need to master this tool, this skill. Okay, so here we have the most recent common ancestor. I am not writing now. Sorry about that. The most recent common ancestor at number 1. And then we have A, B, and C have a common ancestor at number 2. Then they branch and evolve again. So from number 2, we'll have species A, which evolved. And then from the common ancestor number 3, we have B and C. B and C are more closely related to each other than to A. And A, B, and C are more closely related to each other compared to D, E, F, G, and H. Now let's do some examples and let's apply what we have learned, the skill that we've learned in the previous 2 slides, to a specific example. Now this will look very familiar to you because it is grade 11 work, plant diversity. Remember that when you are looking at a question, it's very important for you to read the stem. This over here is the stem. It's very important that we read the stem of the question and we analyze the diagram and we observe, we see what can we observe from this diagram. What is this diagram telling us? It will help us to answer the questions. Below is a common phylogenetic tree showing the evolutionary development of plants. So let's apply what we know before we look at all the other things that look a bit strange here. Here we have the common ancestor again. Right, that is the common ancestor, the origin of plants. Then we see that they evolved and they changed, and they actually show you here that this characteristic that this is the origin of plants, when they evolved and they have the new common ancestor over here, they're adding a characteristic, early vascular plants. Then here's another branch, so that's the point at where it branches, that is the most recent common ancestor. So we have that point over there. And then we have another point over here where the angiosperms branch off from the gymnosperms. So you can see that we are now looking at this vertically. So from the bottom of the most recent common ancestor going up. Now I want to draw to your attention to something that's fairly new here. We are looking at this where the asterisk is. This is the scale. Now I want to take you back to, I think, Monday when Susan did graphs with you. It's very important that you have a ruler when you go into your exam. You cannot just eyeball this one and say that this is the region or this is the number. If you have to read off, you're going to have to, this is the scale, you're going to have to read off maybe somewhere in between there. You cannot just eyeball and say that that is 250 unless you have a ruler. So let's look at the scale. It's showing you the Paleozoic era, the Mesozoic era, and the Cenozoic era. And I wanted to bring to your attention that 0 over here is the present day. And 500 over here is the past. I'm going to write it in here for you. It's present, and this is the past. Okay? So that means that, let's look at this organism, for example. Let's look at a group of green algae. It is still, the line is carrying on until 0. When you are reading a phylogenetic tree and the line of the organism that you are following, if it's reaching to the 0, that is showing you that it's the present day and that organism still exists. I will show you in another example what happens when the species is extinct. So here we have, this one is still in existence, and that's 0, and then you read it. Please take note, when you're reading this, you are reading from 0 to 500. We'll get into more examples later, but I just want to show you that we are reading it going down. Then the next thing I want to draw your attention to, this label here, millions of years ago, right? It's MYA. So here, let's have a look at this. If I have to ask you, when did the most recent common ancestor, when was the most recent common ancestor present for this particular phylogenetic tree? Then you would go here to the origin of plants. You would take your ruler and you would read off here. It's about 480 million years ago. We also can write it as MYA. When we are explaining millions of years ago, we are saying that this existed a long, long time ago. It's not just million years. Million years is, for example, if I have to ask you, for how many years was this species in existence? So if I have to ask you for how many years has the angiosperms been in existence, then you would read off from this point, right? You go all the way across, and then you will see here that it's from about 110 million years ago to present, okay? So then you would count it as 110 million years. That's how long they've been in existence. But we will go through that in more detail in the next slide. What is the phylogenetic tree? This is a common question that they could ask you. Or they could ask you, what does this diagram represent? Which is a phylogenetic tree. But this is asking you for the definition. A phylogenetic tree is a diagrammatic representation of all the evolutionary relationships between species. Now they're asking you specific questions. Which are the youngest vascular plants? Okay, now remember the key word here is youngest. Youngest. So if we are asking for something that's the youngest, it would be closer to the present. So the present is here at 0. And which one, let's go down to the vascular plants. We see here that these are the early vascular plants. So this characteristics of vascular plants originated here from this branch. So then these organisms still had vascular. And then here, this is the most recent one, that the recent branch of the vascular, which are the angiosperms. Then we have the oldest vascular plants. So now we have to go more back into the past, going lower down into our phylogenetic tree. And we see here that at this point here, the early vascular plant, that branch, we have this branch over here. This is where the earlier vascular plants were. And we have the ferns and the horsetails. Let's see if I was right. Yes, that is correct. Okay, so you just go back to your diagram and you try to figure out what they want you to answer. In which era did most of the plant groups evolve? So, where is that point of evolution? That point of evolving, that point of changing is at each, from the common ancestor where it branches. That branch is where the evolution is occurring or where the evolving is occurring. Okay, so here we have a common ancestor. Here we have a common ancestor, and here, so we already have 1, 2, 3 points where it branched. And here's the other fourth one, but that's a little bit more recent. So majority of them is occurring in the Paleozoic era. Okay, so you just count how many were that had the branching, and then we get to the Paleozoic era. Right, the next question. Study the diagram of the rhinoceros group shown below. Once again, we're going to analyze the diagram before we go to the questions. Okay, I know I'm spending a lot of time on this, but it's very important. And like I explained to you, it's a skill that you need to know and you need to master. So this one is a little bit different. This one is now showing you that it's coming from the common ancestor, yes, to the present, but it's a horizontal one, like the first example that we did. Okay, so just to remind you, we are reading it, yes, we read it from 0 to, we are going to read it now from the ancestor to the present day. Okay, but we must take note that it's going, the scale is going from 0 all the way through to 50. Okay, let's have a look at this. So I'm going to do the dots again for the point of the common ancestor. So there, that is the most recent common ancestor of A, B, C, D, E, F, and G. Then it reaches this point and then it branches, and when it branches off, they are going to evolve, they are going to change, and they're going to develop into new species. And then we have a new common ancestor over here, and so forth. Okay? Now, I want to bring your attention to a few other things. You can see here that this phylogenetic tree is a little bit different compared to the one on the plants. How so? In this one, it's showing you the line is ending somewhere in the middle of this graph or this tree diagram. It's not going all the way to the end, to the 0, which is, remember, I'm going to remind you, it is the present. So it's not going all the way to the end, not all of them. We're going to discuss why, but let's go through some questions. This is true and false. State whether the following statements regarding the diagram are true or false. The diagram is called a phylogenetic tree. Is it true or false? I'm going to use, yes, it is true. It's showing you all the reasons why it is a phylogenetic tree. Okay, so I'm going to put T for true. We're going to see later if we're correct. All species have a horn. That's number 2. Do all species have a horn? Let's have a look. This one, A, doesn't have a horn. B doesn't have a horn. And then we have C, D, E, F, and G that have horns. Therefore, this answer is false because they do not all have a horn. B became extinct before A. So now you go to the diagram and you identify where B is and where E is. Okay. Now, this is what I wanted to show you. Let's look at B. B originated here. So you take the beginning of the line. That is when it evolved. Okay? That's when it came into existence. When the line ends, that is the line that indicates to you when it became extinct. Okay, so this line, you can see it's floating in the middle here. That is when it became extinct. So let's look at A. This is where it when it came into existence, and this one over here is when it became extinct, died out, gone, right? The species does not exist anymore. So they want to know, the question is asking, B became extinct before A. So this one over here, that is B. It became extinct, let's say round about, once again use your ruler. I don't have a ruler here to use, but it's round about there, which is 32 million years ago. And A became extinct about 48 million years ago. Therefore, this is false. Because A actually became extinct before B. E is not extinct. So let's go to E. This is number E. And we can see here that the line, this is where it came into existence. And the line over here is at the 0, which means that it is still present in today's times. Therefore, this answer is true. F and G share a more recent common ancestor than E and F. Let's look at F and G. They share a more recent common ancestor, and their common ancestor is this one. Yes, they do share a recent common ancestor. And compared to E and F. So if we look at E and we look at F, we see here that that was the last common ancestor over there. So they do not share the same common ancestor. And F and G share the same common ancestor. So this one is true. D was present and living on Earth for more than 10 million years. So let's look at D. Here's D, and now we're going to count. So now so what I do is I use my ruler, okay? And I first put a dot for when it came into existence and a dot when it became extinct. And I draw my lines going up, and I draw another line going up over there. And this line is a bit wonky, but it's actually meant to be on this line. And if you count over there, it is just short of 10 million years. It's actually 8 million years. So the point I'm looking at is over here and over here. And it is 8 million years ago. Therefore, this answer is false because it's telling us that it's been on Earth for 10 million years. Okay, so we can see that our answers were correct, right? And now we can move on. Another question. This one's about the flightless birds. I'm going to go quickly through this one because Susan actually explained this in a lot of detail when she did biogeography and continental drift. The diagram below represents the evolution of the flightless birds and the continents on which they exist at present. So here we have, once again, we have the most common ancestor. Can you see? Just automatically, I go to analyzing the diagram, writing on the diagram. What can I already observe and conclude from this diagram? Right, so we have the most common ancestor of the flightless bird over there, and here at the bottom they are telling me that the bird species on which continent they are found today. Let's go through the questions. Identify the type of diagram shown. Phylogenetic tree. Right, it's a phylogenetic tree. Have you noticed that they almost always ask this question? Okay, you need to be able to identify that this is also a phylogenetic tree. Yes, it looks a bit different. This one doesn't have a scale, but it is still a phylogenetic tree. Identify the type of diagram shown. Phylogenetic tree, sorry, we did that one. Number 2, name the 2 species that share the most recent common ancestor. Remember I told you, most recent common ancestor? You look at the point where they shared, the point where they have the most recent branching off. And for this one, it's at this point, therefore it's the emu and the cassowary. Number 3, use information in the diagram to describe how biogeography supports the theory of evolution. So Susan did this with you, but to quickly remind you, they all shared a most recent common ancestor, right? Then due to continental drift, the population split. They split into 2 due to continental drift. And then those populations, they changed and they evolved independently of each other due to environmental conditions. They changed and they evolved into new species. Okay, that's in short what was happening there. Number 4, describe how it can be proven that ostriches and rheas are different species. Now, for this one, I want to draw your attention to remind you about population ecology, which you did in grade 11. Okay, in population ecology, you define the terms species and population. So one key word in the definition of a species is that the offspring, they must be able to interbreed and produce fertile offspring. So the best way to check that these two are actually different is that if you give them an opportunity to interbreed, if they are successful, they will produce infertile offspring, or even if they can't, this will show that they are different species. Another way is to collect DNA information and then to analyze it, and then you will see that they are different species. Right, another question on phylogenetic trees. As you can see, we are spending lots of time over here. It's very important that we master the skill. Study the diagram below. So here, it's a bit loaded. It looks a bit hectic and a bit loaded with all of these different words. But don't worry, we are going to unpack them next year when you do grade 12. But for now, it's important that we look at the skill. It doesn't matter what they give you here. You don't have to know what any of these things mean in order to still be able to answer the question. Okay, you're going to be super when you do it next year. So here we have the scale once again. I'm going to draw your attention to present, and then 6 million years ago over there is the past. Okay, and then we have the hominin ancestor over here. There we go at that point, and then we're starting off with our branching. Okay, we're not going, that is basically what this diagram is telling us. Let's go through the questions. What is this type of diagram called? Once again, by now you should know it. Phylogenetic tree. How many of each of the following are represented in the diagram? This is the word genera that I asked you to remember. Genera is the plural of genus. Okay? So what you do here is you go to your diagram and you count. You look at the first word. In the double word of the scientific name, you look at the first word. That is the genus. And you count how many of them are different. Okay? So let's count. We have the first one I see here is Ardipithecus. That's 1. Then the next one I see is Australopithecus. That is 2. Just want to put Ardipithecus here so you can see. Then I see a third one, Paranthropus. Okay. Why did I skip Australopithecus over here around the side? Because they are not different, right? Those are the same. So that's the same genera. They belong to the same genus. Then the fourth one, I have Homo. Right, and then I have the fifth one, which is Pan. Therefore, I have 5 genera. How many Australopithecus species are there? Now I count all of them that have the name Australopithecus. So we have 1, 2, 3, and 4. Let's see if we're correct. Yes, we are correct. Okay, so that is how you do it. Name one species that have Paranthropus arthropus as a common ancestor. So here we need to go to Paranthropus arthropus. Here it is over here, and then you can see that this is, they're telling you that this is the common ancestor, right? So essentially they're asking you who are the descendants of this common ancestor. And we have over here, we have Paranthropus robustus and we have Paranthropus borsaya, and you can also write them as P. robustus or P. borsaya. Please take note, like I mentioned earlier, that the first word, the first letter of the first word is capital. Okay, and then the second word, the first letter is a small letter. Number 4, when did Ardipithecus ramidus become extinct? Ardipithecus ramidus. So we go to Ardipithecus ramidus. Here it is. When did it become extinct? Now remember, we are reading it from the bottom up. So we are going from this point to that point. So it came into existence here and it became extinct here. We draw a line going across, and you will see there that we'll get, I'm going to go right across to there, and you'll see that it's about 4.3 to 4.5 million years ago. When did Homo erectus first appear? It's 1 million years ago. Sorry, I went too fast, but let me show you how to do it. We go to Homo erectus. Homo erectus is over here, and we draw a line at the beginning when it just came into existence, and we draw a line right across. Please use your ruler, not like me who's free handing it here, and you'll get 1 million years ago. Name the hominid species that existed at the same time as Homo sapiens. It's the Homo neanderthalensis, yes, because here's Homo sapien, and you can see that it was present. It's not present anymore, it became extinct, but they were actually living alongside the Homo sapiens, the Homo neanderthalensis. That brings us to the end of phylogenetic trees. I'm going to spend some time discussing evolution in present times with you, but I'm just reminding you once again that the phylogenetic tree is very important for you to remember and for you to master, for the skill that you need to master for next year. Right, so what do we already know about evolution in present times? Or what do you already know about viruses and bacteria? This is what we're actually talking about here. We are going to be discussing how viruses and bacteria play a role in evolution. So from grade 11, you have already learned that rapidly reproducing organisms such as viruses and bacteria allow scientists to study how species change in response to environmental factors. Pathogens evolve quickly because there is lots of natural variation amongst them. Mutations occur more often in rapidly reproducing organisms. Okay, so essentially what we're saying here is that pathogens, all these disease-causing organisms, they all have natural variation. Okay, some of them are resistant to antibiotics, some of them aren't. So what essentially happens is that every time an antibiotic is administered, then the ones that are resistant survive and they reproduce. The ones that are non-resistant will die. And eventually, every time you apply that same antibiotic, they keep on reproducing and they keep on mutating. And that's how they are evolving. So let's look at some common examples that you may know. And you should know all of these, okay? But I'm quickly going to go through them. The Anopheles mosquito is the one that's involved with malaria. At the moment, malaria mosquitoes have actually become resistant to insecticides. Then we have the TB. TB bacteria has become resistant to TB drugs, and because of mutations. Because when they're evolving, they also mutate. So now those mutations are making them resistant to the antibiotics. Then we have, or the drugs are used. Then HIV becomes resistant to the ARVs, and then the one that we are all living with at the moment. We are living with this at the moment, the coronavirus, which we are still currently living with and we are still learning about. But the main thing that we can already see is that we have now emerged with different variants, and that is how they are mutating and reproducing and surviving. Let's do a question. A species of bacterium had two strains, strain 1 and strain 2. A patient infected by this bacterium was treated by an antibiotic that was taken daily for 5 weeks. To determine the effectiveness of the antibiotic, the investigator monitored the patient by measuring the number of each bacterial strain present in the blood samples on a weekly basis. Okay, notice that I am underlining the keywords over here. Then we look at the graph below, which represents the results obtained. Also, recapping you to what Susan did with you, please take note that when you have two sets of line graphs on one set of axes, then when you have two sets on one set of axes, you are going to draw them, you're going to use a key. Okay, you're going to use a key, and you have the dotted line and you have the solid line. Okay, so that is I'm just reminding you about that so that you can see the difference between the two when we are reading them off. Right, let's go into the questions quickly. What was the highest number of strain 2 bacteria recorded for the patient? Right, so we go to strain 2, and then we have at this point over here, we have the highest bacteria recorded. It's there. Sorry, not that one. It is this one. It is strain 2, which is the solid line. It is 500 centimeters cubed. Please note that I'm getting the unit from here. Okay, centimeters cubed of blood. The number of strain 1 bacteria at the end of 3 weeks. So we go here to 3 weeks. You use your ruler and you draw up until you get here to the dotted line, the point of the dotted line, and you read off over there. It is 300 centimeters cubed of blood. Number 2, explain the trend of strain 1 in the first week. When we are asked to explain a trend, then they want you to tell them what is happening and what was the cause of that. Okay, so if we look in strain 1, which is the dotted line, from year to year in the first week, we see that it is decreasing. Okay, the number of bacteria decreased since they were killed or destroyed by antibiotics. Okay, so that is the cause. They were killed. So what happened was that it decreased, and the cause was that they were killed or destroyed by antibiotics. If they ask you to describe a trend, then you would just say that it decreased. The number decreased. Okay, you just describing to them what happened in that particular section. State 3 factors that the investigator could have kept constant in this investigation. Constant factors need to be there to ensure validity of your investigation. What makes your investigation fair? What makes your investigation valid? That is what what they are wanting you to say over here. Okay, so which are the factors that we are going to be keeping constant? Same instrument must be used. Samples must be taken at the same time of the day. Same person must take the measurements. Same amount of blood must be sampled, and the same method of taking the antibiotics. Those are all constant variables, and they ensure validity and fairness. Right, we have time for another activity. Let's move forward. This activity looks a bit loaded, okay? It looks a bit hectic, but we're going through it slowly. And this is what you need to do when you get a question like this in the exam. I also want to bring to your attention that this question, phylogenetic trees and investigations on evolution in present times, those are things that the examiners love to ask. They love to ask it. And it's not really things that you can study, but there are skills that you need to acquire. And the best thing to do in this section is practice. Practice, practice, practice. Okay, so let's read. Read with understanding, read actively, underline the important things. The E. coli bacterium lives in the intestines of pigs where they reproduce rapidly. Certain strains of E. coli cause diarrhea in young pigs. Scientists carried out an investigation using 100 piglets to determine the resistance of E. coli in 2 antibiotics, A and B. The scientists injected the piglet with antibiotic A and antibiotic B. They took a sample of E. coli from the intestines of each piglet a week later and placed them in separate petri dishes, allowed the bacteria to grow for 24 hours, added antibiotic A to one petri dish and antibiotic B to the other, measured the growth of the bacteria in each petri dish after 24 hours, used the growth measurement as an indication of the resistance of the bacteria to each antibiotic, repeated the process over a period of 6 months, calculated the average percentage resistance to both antibiotics. Now, I want to bring your attention that this over here, what the scientists did, that is the method. Okay? And then I want to show you that here we have a sentence over here. This sentence is the aim. Okay? The aim of the investigation. I'm not sure if you can see there. This part is the method, and this sentence over here, this is the aim. Okay? So the aim is if you see the word to determine, the words to determine, that will indicate to you that it is the aim of the investigation. And the aim of the investigation is where you will find the independent and the dependent variable. Okay, so I'm just reminding you about that. And then I want to draw to your attention to these last bullets over here, which are saying that they repeated the investigation and they calculated the average. This is telling us this is the reliability. This makes the investigation more reliable. Right, going through the questions quickly. I've actually answered a lot of them, and I see the clock is also going. Identify the independent variable in this investigation. It is the type of antibiotic. Yes, it's the type of antibiotic. I quickly want to go back to show you why I say that. Because over here it's showing you that these two antibiotics were administered. So the type of antibiotic was administered. And then they checked the resistance of the E. coli against that. Then we go to the identify two factors that should be kept constant. These are during your investigation. Remember I said they're ensuring validity, environmental conditions, amount of antibiotic, concentration of antibiotic, okay, just to name a few. You will see that many of them you will get actually from the method when you see what they were doing. State two ways in which the scientists ensured the reliability of the investigation, and we spoke about that. It was done over a period of 6 months. They took measurements, calculated an average, and they used a large sample size of 100, or you can just say 100 piglets. Which antibiotic will you recommend for controlling E. coli in piglets? We would use antibiotic B. Okay, so how did we figure this out? I wanted to show you before we do that. You go to the graph, okay, and you see there what actually happened here. Antibiotic B, you read the average resistance percentage. It was constantly lower compared to antibiotic A. So when they ask you to support your answer, you say exactly that. The average percentage resistance of E. coli bacteria to antibiotic B is lower than its resistance to antibiotic A. Therefore, more E. coli bacteria die in the presence of antibiotic B. Right, that's all we have. That's all we have time for today. I just put this question in just to show you that they can also give you an extract. Okay, like this. You need to learn how to read with understanding and underline your keywords. It's very, very important to do that. And to read everything here, they give you a case study, they give you a method of what scientists did, and they give you some results over here. So it's very important that you go through all of these you read through all of these actively. And I just want to remind you again, please, phylogenetic trees is a skill that you need to master, and it's a question that the examiners love. And so evolution in present times, they love to ask investigations in this topic. So please make sure you know how to do it. Thank you for watching this lesson. Thank you for listening to us. Please remember to sanitize and to wear your mask and adhere to all your COVID-19 protocols, and enjoy your holidays. Thank you.